Next agenda is our keynote session. This is a talk by Dr. Mrinal Patanik entitled Understanding Monocyte-Related Disorders, including Epigenetics and Genomics. Dr. Patanik is a colleague of mine. He's a hematologist and the lead for our acute leukemia myeloid neoplasm disease-oriented group. He is also the enterprise-wide Mayo Clinic Cancer Center co-lead in cancer individualized medicine. He leads our group in the epigenomics, epigenetics, and also has a special interest in bone marrow failure syndromes. He regrets he cannot be here today due to a conflict in his schedule. However, he has made a recording and will try to zoom in the last 10 minutes of this session for a Q&A session. Can we play the video? Okay.
And then dwell into the ontogenic origins of Langerhans histiocytosis in particular. So, ladies and gentlemen, thank you for the opportunity to present at this meeting. I'm sorry that I cannot be there in person, but I hope this presentation will help stimulate you in terms of some of the overlaps that we have in monocytic neoplasms and disorders of histiocytes and macrophages. So these are my conflicts of interest, and I will not be speaking about any off-label drug recommendations. The objectives today are to really define human monopoiesis, describe myeloid neoplasms with monocytosis, discuss the role of tissue-resident macrophages and monocyte-derived macrophages, and then dwell into the ontogenic origins of Langerhans histiocytosis in particular. So if you look at this slide, this is an elaborate schema of hematopoiesis in humans. And I would like to draw your attention to monopoiesis. So you have the multipotent progenitor cell, which then gives rise to megakaryoblast, proerythroblast, and myeloblasts. And the myeloblasts can then differentiate into different morphological subtypes, but importantly here is the monoblast that forms a promonocyte that can then differentiate into monocytes, which can then eventually result in further maturation into myeloid dendritic cells and the monocyte-derived macrophages. And so there is an elaborate epigenetic post-mitotic differentiation schema. And the monocyte is able to enter into different niches and spaces, whether this is in the bone marrow or various tissue and viscera, where it forms a monocyte-derived macrophage and then plays a very important functional role as an antigen-presenting cell. And these monocytes are of different types. And as you can see over here, that monocytes broadly, and of course, this classification continues to evolve, but based on flow cytometric expression of CD14 and CD16, they can be divided into classical monocytes, or MO1, which are brightly positive for CD14 and negative for CD16. The MO2 and MO3, which form the non-classical component, with MO2 being intermediate with dim expression of both 14 and 16, and the MO3, which are negative for CD14 and positive for CD16. And the macrophages themselves, depending upon their exposure to various chemokines, cytokines, can broadly evolve into two different or distinct phenotypes. The M1 macrophage, which is really bactericidal, inflammatory, involved in immunostimulation, and the M2, which plays a better role in scavenging with tissue repair, matrix remodeling, angiogenesis, and immune suppression. And if you look at this ontogenic, cascade, it's this progenitor cell that forms these myeloid dendritic cells, and these monocytes, based on colony stimulating factors and their receptors 1R, 2R, 3R, that form these classical monocytes, largely driven by CSF1R receptor expression, the intermediate monocytes, and then the non-classical or MO3 fraction. And then based on, exposure largely to interferon alpha and then IL-4, IL-13, these can become tissue resident macrophages of monocyte origin, which then can form the M1 or inflammatory macrophages, and the M2 or the fibrotic macrophages. And while this is going on, it's also important to note that there are tissue resident macrophages, which have a embryonic origin from the fetal yolk sac. They have different names depending on the organ where they populate during embryogenesis, and they have replicative capacity. So in the bone, these are called as osteoclasts. In the central nervous system, they're called as microglia. Connective tissue, they're called histiocytes. In the placental chorionic villi, they're called as Hofbauer cells. In the kidney, they're mesangial cells. In the liver, they're Kupffer cells. Then pulmonary alveolar macrophages in the lung, epidermal and dermal macrophages in the skin, and then the red and white pulp macrophages in the spleen. And this review that was published in Cell beautifully highlights, the during embryogenesis from the yolk sac, you have these tissue resident or RTMs that originate, they populate into the central nervous system where they form microglia. They enter into the lungs where they form pulmonary alveolar macrophages, and then they're present in the intestine where they're called as the intestinal macrophages. And while this occurs, there is a dynamic equilibrium that is also regulated or maintained by monocyte-derived macrophages. And you can see these pie charts that show over time, for example, in the central nervous system, most of the microglia are embryonic with a small fraction being monocyte-derived, whereas this increases in the lungs and in the intestines, into adulthood. This is now largely being monocyte-derived. And what is the homeostatic cues for maintenance and responsibilities? As you can see, the microglia, they really play an important role in pruning damaged myelin sheets. They're maintained by astrocytes secreting IL-3, IL-33, neurons in glial cells secreting IL-34 and MCSF. In the lungs, they play an important role in recycling excess surfactant, and there are disorders where there are mutations of GM-CSF or antibodies to GM-CSF, or macrophage monocyte defects such as GATA2 haploinsufficiency, where excessive surfactant can accumulate in the pulmonary alveoli, a condition called as pulmonary alveolar proteinosis. And these are also maintained by the IL-34 GM-CSF axis. In the heart, the cardiac macrophages protect blood vessels and they facilitate electrical conduction through cardiomyocytes. The Kupffer cells eliminate senescent red blood cells and cellular debris. The lamina propria macrophages in the intestine play a major defensive role there, eliminating bacterial pathogens. And osteoclasts are very, very important in reabsorption of bone because of regulation with the rank ligand and MCSF, and abnormalities in this pathway can lead to excessive bone deposition and pathological fractures, a condition called as osteopetrosis. And this balance is altered in disease. So if you look at the resident tissue macrophages, as we mentioned, microglia, pulmonalveolar macrophages, the LPMs in the intestine, Kupffer cells. Moment you get either degeneration in the form of Alzheimer's disease, chronic inflammation in the form of cirrhosis, Hep B, Hep C, or hepatocellular carcinoma. In the lungs, chronic infections or even acute catastrophes like COVID-19, inflammatory bowel disease, and then atherosclerosis. There is a larger contribution that's coming in from the monocyte-derived macrophages or the MOMACs. And, these tissues infiltrate largely driven by chemokine called a CCR7, which helps this transmigration process. And, when you look at the transcriptomic profile, it's important to note that when alveolar macrophages, Kupffer cells, or LPMs have been analyzed at a steady state, there is a contribution from monocyte-derived resident tissue macrophages and then embryonic resident tissue macrophages, which you can see here, again, dominated in the lungs, to some extent in the liver, but in the intestines by adulthood, they become the dominant source, and they have expression of certain genes. But when there is inflammation, the monocyte-derived MOMACs, their contribution clearly increases. This is the light blue that is added to this pie chart. And what drives a lot of this is the overexpression of TREM2, which is the triggering receptor exposure on myeloid cells too. And TREM2, has a elaborate signaling network through the PI3 kinase mTOR pathway. It leads to increased protein synthesis, proliferation, clustering of cells, also leads to autophagy. And, there's data showing that TREM2 polymorphisms, especially R47H, is a risk allele for development of Alzheimer's disease. Now, the dendritic cells are not to be left behind. We have the conventional dendritic cells that are positive for CD141, BDCA1 and BDCA3, and then the plasmacytoid dendritic cells that express CD123, BDCA2, and BDCA4. And then depending upon their location, they have, again, elaborate functionality in the blood, the skin, the lungs, the spleen, and the gut. And this highlights, some of the surface marker expression that can help differentiate the origins and determinants of dendritic cell function. So, in our area of expertise, we're very interested in monocytosis, and there is a plethora of, myeloid neoplasms that can present with monocytosis, including the prototype in adults, which is chronic myelomonocytic leukemia. You can have myeloproliferative neoplasms in monocytosis. The pediatric counterpart is juvenile myelomonocytic leukemia. Molecularly derived entities, especially due to rearrangements of the gene PDGFR beta, the rare CML P190 isoform, and then, of course, the acute leukemias, the old FABM4 and M5A and B, which are the myelomonocytic and monoblastic leukemias. So CMML is a disease of aging. It's an overlapping neoplasm with features of MDS and MPN. The median age of onset is 73, although the incidence is similar to myelofibrosis. Because 15 to 30% of patients transform to AML over 3 to 5 years, the prevalence is much lower. So technically, it's an orphan disease. The median survival is less than 3 years, and it has a high rate of AML transformation. And this is a prototypic CMML patient. You can see in the blood, there are these immature monocytes that are called as promonocytes. In this particular patient, they're dysplastic granulocytes. The bone marrow aspirates are typically hypercellular with granulocytic dysplasia, which may or may not be present. And the core biopsies are hypercellular. The stain that we often use to show dysplasia is the dual esterase stain, where you can see these dysplastic monocytes taking up, the butyrate esterase as well as chloroacetate esterase. And about 30% of patients have nodules composed of plasmacytoid dendritic cells. You can see this on the H&E stain. And this is higher power view showing the spindle shape morphology. and they're brightly positive for CD123, CD303, and can be positive for TCL1. And there's a fair number of patients that may also harbor another spindle cell population that's more clustered, aberrantly expresses CD2 and CD25, and will be positive for CD117 and tryptase, which is systemic mastocytosis, giving rise to the most common associated hematological neoplasm with SM, which is CMML. And, this is adding to the literature that has been published by several of you in the room. And this is collaboration with Gaurav and Ron, where, we had a patient who had Erdheim-Chester's disease. You can see, the skin rash in A and B, the Tauten giant cells in the dermis, and C, the immunohistochemistry for CD163, which really is a macrophage marker that can be seen in monocytes and transition to macrophages too. The dual esterase stain, and then the technetium uptake around the knee joints, and sequencing, showed that there was a significant overlap in shared mutations, giving rise to the important viewpoint that there could be shared ontogenic evolutionary patterns that we need to be aware of. And we've seen this in CMML, for example. This is a paper we published a few years ago where, in 98%, when CMML transforms into a blast phase disease, it's usually in the form of AML, but rarely it can transform into blastic plasmacytoid dendritic cell neoplasm. And so you can see over here, this is at the CMML time point where there are these promonocytes in the blood. We see these plasmacytoid dendritic cell nodules that are TCL1A and CD123 positive. And then the transformation to BPDCN a few years later because of copy number alterations involving copy neutral loss of heterozygosity of the retinoblastoma 1 gene. And the marrow is now packed with these blasts that have a hand mirror morphological appearance, as you can see an F, suggestive of BPDCN. And when you see the stain here now, the TCL1 and CD123 is uniformly positive. And, these patients don't do well even with targeted therapies such as tagraxofusp that target CD123 with the diphtheria toxin. Now, CMML diagnosis is evolving. And in 2022, there were two classification schema that were developed, which had slightly divergent views, but they both agreed that the monocyte count threshold for CMML can be lowered to 500. So in the fourth edition of the WHO, the cutoff used to be 1,000. As long as the monocytes are more than 10% of the differential, the blasts and promonocytes are less than 20%. And we rule out these myeloid lymphoid neoplasms with tyrosine kinase fusions. In the population that has a monocyte count between 0.5 to 1, it's important to show some form of clonality, and whether this is in the form of a cytogenetic abnormality or somatic mutations involving ASXL1, TET2, SRSF2, the presence of those with the absence of reactive causes of monocytosis are very helpful in increasing the confidence of calling this as CMML. And some of us still call this 0.5 to 1 as oligomonocytic CMML, which I think is in the spectrum. When you look at CMML genomics, the most common mutant gene is TET2, which is a dioxygenase that converts 5-methylcytosine to hydroxymethyl, followed by ASXL1, which modifies critical histone configurations, then SRSF2, which is a gene that controls pre-mRNA splicing. And the proliferative CMML subset is really enriched in signal mutations, most of them being RAS pathway, with a small subset having JAK2 mutations. And when you look at the clonal hierarchy, the initial aberrations are almost always TET mutations, which are then associated with either biallelic TET mutations or TET mutations with copy number alterations or TET with SRSF2. And then depending upon the subsequent acquisition of somatic changes, It shapes either a dysplastic or a proliferative phenotype, which over time can evolve into acute myeloid leukemia. And just to give you how complex some of these can be, this is a 58-year-old gentleman who presented with leukocytosis and monocytosis with anemia and thrombocytopenia. A bone marrow biopsy was hypercellular with 10 to 15% blasts. The megakaryocytes were increased with atypia, and there were large hyper and occasional hyperlobated forms with focal clusters. There was grade 1 reticulin fibrosis. The blasts were clearly myeloid, and the karyotype revealed deletion 7 and deletion 20q in 20 metaphases. And so the patient was diagnosed as CMML2 and sent to us for evaluation. When you look at the blood smear, in the top left-hand panel, you can see the promonocytes. But when you looked at the bone marrow, the reticulate stain showed fairly extensive, 1 to 2 plus reticulin fibrosis. The aspirate had clearly, an excess of blasts, and you can see over here a clear myeloblast. And there were occasional islands that were positive for CD123, but negative for CD303. And when we did sequencing, we found there were somatic mutations involving ASXL1, MIPL with a VAF of 57%, SRSF2, and dual TET2 mutations. And the presence of a non-JAK2 MPN driver in CMML really raises the question as to whether or not this is CMML. And so a very good adjunct that has been added to the WHO criteria for diagnosis of CMML is monocyte repartitioning flow cytometry. And so you can see in this case, we did flow cytometry. This is a control, a CMML patient, where the MO1 fraction is greater than 94%, which is highly sensitive and specific for CMML diagnosis. And this is the patient who was thought to have CMML, where MIPL is a driver, the MO1 fraction is less than 94%. And when you look at the bone marrow, you can see that these, hyperlobate megakaryocytes actually tend to cluster. And so this really is not CMML, but a patient of MPN with monocytosis. And when you look at the data, from Mayo Clinic, Dr. Teferi has shown that monocyte counts that are increased are an adverse prognosticator in primary myelofibrosis. They tend to occur at an older age with a higher white count, lower platelet counts, and a higher frequency of circulating blasts. There is a trend towards a higher frequency of ASXL1 mutations, but clear survival difference, especially if your absolute monocyte count is greater than 3,000. You can see that median survival is less than 2 years. And this also extends into polycythemia vera. And this is data from the P-Vera cohort from Mayo, where the red line, are patients with a monocyte count greater than 1,000 who had a shortened median overall survival, but it did not impact their myelofibrosis-free survival or their leukemia-free survival. So the mechanism by which it reduces overall survival is independent of leukemic transformation or myelofibrosis transformation. And this segues into the ontogeny of histocytic neoplasms, which share significant overlap. This is a smattering of slides from patients with histiocytosis. Panel A is Langerhans cells histocytosis, where you can see that these histocytic cells are positive for CD207. They most often express the BRAF V600E point mutation, and under ultrastructural evidence, they show these Burbeck granules. In panel B, which is a liver specimen, again, you can see morphologically the histocytic infiltrate with these cells being positive for CD207. In panel C, this is a neurodegeneration specimen where you can see the BRAF stain, the CD163 positivity, and the P2RY12 positivity. Whereas slide D shows a case of a mixed LCH with juvenile xanthogranulomatosis, where you can see the giant cells as well as the histocytes in D1. In D2 and D3, there are two different populations of cells, one being the histiocytes, which are CD207 positive, and then the granulomatous cells, which are CD68 positive. And this review in the New England Journal of Medicine very nicely highlights some of the physiological Langerhans cells and the epidermal LC ontogeny and function. So as we've reviewed, the bone marrow stem cells can give rise to monocytes as well as dendritic cell precursors, whereas the fetal yolk sac gives rise to these resident tissue macrophages. And you can see that the epidermal Langerhans cells are originating from the fetal liver yolk sac progenitors, whereas the dermal dendritic cells originate from the hematopoietic compartment. And these monocytes, they can transmigrate because of CCR7, which is chemokine 7, and play an important role in terms of antigen presentation, T cell activation, and apoptosis. And some of the mechanistic overlaps that exist are believed to occur because of different stages during differentiation in which somatic mutations are required. So, for example, if they occur in the CD34 positive stem cell progenitor, they lead to high-risk multisystemic LCH. When they occur in committed dendritic cells, precursors or monocytes, they lead to low-risk multisystemic LCH, whereas if they occur in committed dendritic cells or tissue-resident monocytes, they lead to low-risk single-lesion LCH. If they occur in the fetal liver yolk sac, they impact the resident tissue macrophages, and if this involves the epidermis, this leads to self-healing congenital skin LCH, whereas if it comes from the monocyte-derived tissue macrophages, it can, depending on the organ, result in abnormalities in the lung or the liver or in the brain, a neurodegenerative phenomena after systemic disease. And most of these somatic mutations involve the MAP kinase pathway. And we will see that between BRAF and MAP kinase mutations, followed by mutations in other genes, this results in enhanced myeloid differentiation, impaired migration, inhibition of apoptosis, and inflammation. And this is a slide set showing in LCH that more than 60% are BRAF V600E. There are BRAF fusions, BRAF indels, mutations involving MAP2K1, abnormalities in ARAF, ERB3, and even other minor pathways in RAS. And this is very similar to myeloproliferative CMML. So drawing comparisons again, this is a large data set with functional work that we've published in Nature Communications, showing that there's a clear survival difference between proliferative and dysplastic CMML divided by a presenting white count of greater than or less than 13,000. And when you look at the proliferative, which is highlighted in red in this slide, it's largely driven by RAS pathway mutations. And in this particular paper, KRAS, NRAS, Sybil, PTPN11, NF1, and several minor RAS pathway abnormalities could be seen in more than 80 to 90% of patients. We did demonstrate that, RAS cannot be viewed as a binary variable and that there are variant allele fraction thresholds that correlate well with proliferative disease. And this was 30% for NRAS and 20% for RAS pathway. And when we compared paired samples at CMML diagnosis, which is in blue, and at leukemic transformation in 40 patients, we found that the dominant abnormalities were either incremental gains in RAS pathway mutations or acquisition of new RAS pathway mutations concomitant with ASXL1. But genetic events only explained about 48% of all CMML to AML blast transformations, telling you that there's an important role of copy number alterations and epigenetic phenomena that need to be deciphered. Now, clonal hematopoiesis is defined as the acquisition of somatic variants in hematopoietic stem and progenitor cells, which confers increased fitness and has the ability, to select over time and under cell intrinsic and extrinsic selection pressures. This is clearly an age-related phenomena, but there's context-specific clonal hematopoiesis. And so clonal hematopoiesis in patients with bone marrow failure syndromes, with telomere biology disorders, with prior exposure to chemoradiation, has a different spectrum in comparison to age-related clonal hematopoiesis. And clonal hematopoiesis is very much existent in these histiocytic neoplasms. And as you can see in this data set on Erdheim-Chester disease, TET was the most common CH mutation seen, followed by ASXL1, DNMT3A, and then a spew of myeloid mutations. And from a median variant allele fraction perspective, again, TET was the most common. And this is significant because the acquisition of TET mutations at the progenitor cell level does skew enhanced differentiation for the multipotent progenitor cell towards granulocyte monocyte progenitor axis that leads to enhanced monocyte biology. And we were able to publish this year in leukemia using single cell proteogenomics in CMML patients. And as you can see over here, that when we sequenced close to 4,000 single cells from this patient, the the ancestral clone was SRSF2 mutant, acquired a TET mutation, and then the 2 TET SRSF2 clearly was dominant with 3,956 sequence cells carrying these dual TET mutations with the SRSF2. And we are now able to, using the BioLegend cocktail and appropriate spike-ins, highlight what cell types are mutant or wild type, and very accurately map out clonal hierarchies, which I think would be very instructive in histiocytic neoplasms and disorders of macrophage activation. And I would like to finish with the poignant question on whether we can exploit similar targets in these diseases. Genetic targets in CMML have been difficult to capitalize on in comparison to AML and MDS. Mutations in IDH1, 2 are infrequent. P53 is almost never seen. FLT3 is very, very uncommon. And although RAS mutations are common, MEK inhibition, unlike in histiocytic neoplasms, probably because of the high proliferative rate and the off-target toxicity, has not been effective. The trial with trametinib hasn't worked, and there is a cobimetinib trial that is currently accruing. The covalent inhibitors of KRAS G12C that have been approved in lung and colorectal, such as sotorasib and adagrasib, are seen in less than 1% of CMML patients. And although splicing mutations are very common, the splicing inhibitors have really not advanced. And we're hoping to see some promise with the IRAK4 inhibitors or the CLEC1 inhibitors, which are going through early phase testing. And so we carried out this massive project where, firstly, we showed that on RNA sequencing, proliferative and dysplastic CMML clearly have a different transcriptomic profile. The transcriptomic profile is highly enriched in mitotic checkpoint kinase regulators such as PLK1, WEE1, and that, the VAF directly or linearly correlates with the expression of these genes. In particular, PLK1 was of great interest because it was unique to RAS and not seen in JAK2 mutant CMML. And here you can see that, when we knock down NRAS, we decrease PLK1, and when we knock down KRAS, we see the same, versus when we transduce NRAS or KRAS, we drive up PLK1 expression. And we were able to translate this into a preclinical trial first, with NSGS mice using volasertib and onvansertib, which are PLK1 inhibitors, where you can see in the spleen and the bone marrow CD163 and human CD45 showing engraftment of malignant cancer cells and monocyte macrophages in the bone marrow. in the spleen and the bone marrow with good clearing. And then on flow cytometry, you can see that the human CD45 in the drug compared to the vehicle, there's marked clearing. And the same thing happens in the bone marrow and in the spleen. And the spleen volumes were reduced, spleen weight improved, and this has now led to a phase 1 trial at Mayo Clinic MC210807, where we are using onvansertib, an oral highly selective PLK1 inhibitor for patients with relapsed refractory or intolerant proliferative CMML, with the hypothesis that this is going to be effective in RAS mutant. And again, this is more xenograph work showing, in the bone marrow, spleen, effective clearing of cells, and some of the single cell work that we're doing to determine response and resistance mechanisms. We're also focusing on the epigenome, and this is a large data set that we published again in Nature Communications, looking at the oncogenic enhancer elements regulating ASXL1 mutant CMML, using several histone configurations with ChIP-seq, DIP-seq for 5-methyl and hydroxymethylcytosine, and ATAC-seq to look for chromatin accessibility. We've defined permissive promoter chromatin states, looked at gene body hypermethylation, and then looked at ASXL1 mutant specific distal enhancers. And we were able to define these enhancer regions and transcription factors that we have listed over here, and have shown that BRD4 and P300 are very important epigenetic readers for these regions, with P300 being the effector of their changes, and are now in the process of launching a phase 1 trial with Epigenetics using their inhibitor in ASXL1 relapse refractory CMML. So ladies and gentlemen, in summary, myeloid neoplasms with monocytosis can share diverse genetic features, with similar phenotypes, and their ontogenic evolution has to be kept in mind. Things like NGS, flow, and morphology should be used in combination and remain the mainstay in differentiating them. Given the shared pathobiology, important similarities and differences exist between CMML and histiocytosis or macrophage-related disorders that we need to continue to work on, but this should culminate in shared research and drug development, which would be highly beneficial for all. I would like to really thank CCATS, Hematology, SIM, our lab members, and several collaborators, including Eric Solari, Eric Padron, Crispin, Jing Zhang, Klaus Geisler, and the Zapico Fernandez lab. With that, I thank you very much, and we'll be very happy to take questions. Thank you. Ladies and gentlemen, thank you for the opportunity to present.
Getting an echo here. Are we good? Hello. Oh.
And disorders of histiocytes and macrophages. So
Hello?
These are my conflicts of interest, and I will not be speaking about any off-label drug recommendations. The objectives today are to really define human monopoiesis, describe myeloid neoplasms with monocytosis, discuss the role of tissue-resident macrophages and monocyte-derived macrophage.
Okay, I think we are good. Well, that was a very fascinating talk. Thanks, Dr. Patnaik. I think he's joined us on Zoom to take questions. Just want to confirm if we are live. with
Can you
Dr.
hear me? Can you hear me?
Okay. We did say him, but there is echo. Echo. Still there. Are we good? No.
Again, thank you for the opportunity, and I'm sorry I can't be there in person, but happy to take any questions, and just fascinated by the overlap between these monocytic neoplasms and the histiocyte disorders.
Thanks, Dr. Patnaik. We'll take any questions from the audience. It was very interesting, thought-provoking lecture. I think I'll get us started. In the meanwhile, one of the interesting things is we see where these fields overlap is the concomitant myeloid neoplasms, especially CMML, and we have worked on that case together. And when I look at it from histiocytosis aspect, the mortality is high in those cases. The cases of histiocytosis that develop myeloid neoplasm.
Check.
The mortality is pretty high in those cases. So what would be the best way, in your opinion, to study these associations? The CMML is sort of a ECD, the overlap happens with CMML, but CMML per se is not targetable, whereas histiocytosis are pretty targetable using BRAF and MEK inhibitors. So do we think, the question comes is, are there like unique instances? Can they be targeted, or what would be the way to study these prospectively? Because we can't predict who's going to develop those myeloid neoplasms. I don't know if my question makes sense.
Yeah, no, I think it's a parts of your question I couldn't hear clearly, but I think I got the overall. And can you hear me okay?
Yes.
Okay, great. So yes, we've seen this quite frequently, and as you correctly pointed out, CMML and then to some extent myeloproliferative neoplasms tend to overlap, with these entities. And it becomes quite difficult because, as you pointed out, that MEK inhibition may be, firstly, BRAF per se is very, very uncommon in CMML. So most of the time, either you have further evolution in the tissue spaces with BRAF, or these are other RAS pathway mutations that are non-BRAF. But even MEK inhibition, for example, with cobimetinib, we've tried other MEK inhibitors too. Given the high proliferative rate in CMML, not only is it not effective, but it also worsens cytopenias and the toxicity. You can make the cure of disease more grievous than the endurance of the same for those patients. So I feel like as a community, we really need to understand the origins of these conditions, especially when they overlap. And I think with the advances that have been made in single cell sequencing and in spatial omics, combining the two to understand, okay, is this originating from the same stem cell defect? Where is the evolution occurring into a histiocytic morphology versus a monocyte, biology disorder, and trying to understand whether there are mechanisms of targeting both, without necessarily. I feel what we are doing right now is targeting the end spectrum of differentiation rather than trying to understand what are the early vulnerabilities that we could exploit. A lot of these are going to be epigenetic, because if you see, how these monocytes differentiate or how monocytes differentiate into macrophages and histiocytes, these are elaborate histone modifications and methylation changes that we should study, and would be very happy to do that with anyone willing to collaborate with us.
Yeah, because I think that's a fascinating opportunity. These are rare instances, but all of us probably by now are collecting specimens on these cases. And short of just prospectively trying to figure out who's going to develop and take decades to answer, I wonder if there's a concerted effort that could be made for these cases and collaborate maybe with your lab, or if there is interest.
Yeah, I strongly agree with that, Gaurav. I think, with our Mayo Clinic core, the epigenetics core now has the entire spectrum of single cell omics available, from single cell DNA, RNA, attack-seq, multi-omi, including the spatial transcriptomics using the 10x platform. So we should really coordinate and find these cases and try and, ascertain how this is evolving, what are the shared origins, and where we can consider targeting rather than waiting for, trying to hit MEK and other things, which I feel are effective in histiocytosis, but really I'm quite pessimistic on how they will work on the hematopoietic side of things.
Thank you. Any other questions for Dr. Patnaik? Oh, yeah, Dr. Reck.
Hi, Mrinal. This is Karen Reck.
Hi.
I just had a question if you or Grav have any idea about what proportion of ECD patients have an underlying CMML diagnosis.
I think Gaurav might be better. to
Yeah, I
answer
think so. In the two studies, the largest one I think was a collaboration between French cohort and Memorial Sloan Kettering. And then from our own Mayo cohort, concomitant myeloproliferative myelin neoplasms are actually between 10 to 15% of the cases. So that's the percent. And that's why it's, you have a rare disease and then you go to 10% of those, it becomes ultra rare. So that's sort of the prevalence of concomitants. Now, I don't know, we haven't studied the, bone marrow milieu and, how many have some predisposing condition or something that could be harboring CMML in advance. And I don't know how to study that. I don't know if Dr. Patnaik has anything to.
I mean, I'll give you one example, Gaurav. So we saw a patient recently who had CMML, and this was driven by an NRAS mutation with ASXL. And then 2 years into the diagnosis, developed extensive cutaneous lesions that look like urticaria pigmentosa. So we were able to biopsy the skin lesions, sequence the skin, and then sequence the bone marrow CD34 progenitors. And clearly what happened was the skin lesion had the same mutations as the CMML CD34 progenitors, with the exception that it had now acquired KIT D816V as a mast cell differentiator in the skin. And so I feel that, while it's important to work in silos, if we have to make major breakthroughs and discoveries in these rare diseases, coming together and understanding the ontogenic origins. So how much of it is monocyte derived, how much of it is tissue resident derived, how much of it is monocyte resident derived. And I feel that really clonal hierarchies with single cell omics will be the next step.
Yeah, we have another question. Dr. Dave.
Hi, so how how often do you see brain metastasis with CMML? And those cells that enter the brain, are they immunophenotypically altered from the cells that are there systemically? And also, if you do see it with any frequency, has neurodegeneration been observed like we do with, for example, LCH and ECD?
Yeah, great question. Actually, can I just deviate to, I know what you're asking. So in CMML, we almost never see parenchymal disease. It can be rarely leptomeningeal. It usually occurs when there's blast transformation to AML. But I'll highlight your point. I'll take you to clonal hematopoiesis, where there was a very disruptive abstract presented at ASH last year showing that clonal hematopoiesis protects people from Alzheimer's disease. And so we were, and this was Mendelian randomization, biobank data, as well as single cell data. And what really was happening there was the plasticity between macrophages that were TET mutant to go and replenish depleted microglial cells and scavenge neurofibrillary tangles in the brain. That is the hypothesis that drives this. So I think in the brain, there is a lot of monocyte replacement for microglial degenerative changes with aging in clonal hematopoiesis per se. And that's going to be the next frontier to understand how these genetically aberrant macrophages influence microglial function in the CNS.
Thank you. Thanks, Dr. Patnaik, for joining us from wherever you are. I'm sure you're educating other folks. I think you're doing some ASH event right now.
Yeah, I'm in Brazil right now. So thank you for having me, and look forward to collaborating soon.
All right, let's give a big hand, Dr. Patnaik.